This is an exam application tutorial on the topic of protein synthesis. The question for 6 marks is explain how a genetic mutation can result in a faulty enzyme being produced during protein synthesis. So you're going to have to draw on a lot of knowledge here. This is quite a complex and sophisticated question, and it has many levels. Here, you're being asked to discuss the process of protein synthesis. You have to explain what a genetic mutation is and how a genetic mutation can affect the process of protein synthesis to produce a faulty enzyme. An enzyme is a protein, so it's saying a faulty protein. So the translation for this is how is the information in a gene used to make a protein? That's explaining protein synthesis, basically. Then, what is a genetic mutation? So explain how a genetic mutation can result, yada, yada, yada. But you're explaining what a genetic mutation is. And then finally, why can a mutation affect a protein structure? So how does a mutation cause a faulty enzyme to be produced? So be aware in this question, you do not have to explain all of protein synthesis in detail. You do not have to explain the process of transcription and translation in painstaking detail. That would just be a 6-mark question in its own right. Instead, you really have to be clever and you have to summarize the process of protein synthesis. So how can you do that? Well, you can do it in 2 sentences. First sentence, the sequence of bases in a gene determines the mRNA base sequence. So you can see here, this is a section of DNA, and the information on DNA has been copied onto a molecule of mRNA. So basically, the sequence of bases in a gene in DNA determines the sequence of bases in mRNA. If you have an A here, you'll have a U here on mRNA, C, G here for mRNA, A on DNA, U, and so on, T to A, and so on. You can see how the DNA sequence determines the mRNA sequence. So that summarizes transcription. Now for summarizing translation. This determines, in other words, the mRNA base sequence determines the amino acid sequence in a protein or polypeptide, same thing. So if you remember, every 3 bases, which we call a triplet or a codon, will code for one specific amino acid. So the sequence of codons on mRNA will basically determine what amino acids are found in our protein and the order in which they're found. So the sequence of bases in a gene determines the mRNA base sequence here, and this determines the amino acid sequence in a protein here. So now we've addressed how the information in a gene is used to make a protein. Now let's look at what a genetic mutation is. A genetic mutation is a change in the base sequence of a gene. So you can see here's our normal base sequence in our gene, but here we've had a mutation. One of the letters has changed from a G to a C. So we have changed the base sequence, the order of bases, and what base occurs in the gene. So this can change the amino acid sequence of a protein or polypeptide, because if you play this forward, you're going to get a different sequence of bases on mRNA, and now because you have a different sequence of codons, you'll get a different amino acid in our protein. So we're on about 4 marks now. Now we have to deal with the last part of the question. Why can a mutation affect a protein structure? So basically, the sequence of amino acids in a protein affects how this polypeptide chain, this protein chain, folds to make a 3-dimensional structure. This is the normal gene, and it codes for an enzyme with this shape. But if we change the sequence of codons here, and therefore change the amino acid sequence, it won't fold in the same way. So now our protein, or let's say enzyme, has a different shape. It now is faulty. You can see the active site no longer fits the substrate, whereas here it did. So a change in the amino acid sequence of a protein can cause the protein's shape to change. For example, an enzyme's active site may change shape, and in brackets so it can no longer bond with the substrate. So I've dropped in enzyme here because they do talk about enzymes in the question, so it would be unwise not to reference the protein they are talking about in the question.